Hi, good morning, everyone. Let's start the first session of today. The name of the session is Neuroinflammation. We are Yoshi and Matsuya will chair this session. So please.
Okay, good morning. So I would like to introduce the first presentation. It's about evaluation of effectiveness of dolutegravir on neurological outcomes in HTLV1 patients, an open-label, randomized, controlled by pilot study. I'd like to invite here Iris Montano Castellon from Federal University of Bahia, postgraduate program in Medicine and Health, Brazil. Please.
Good morning. My name is Iris. Thanks to the organizers. So today I'm going to talk about this controlled pilot study. This is a pilot study, and for the evaluation of the effectiveness of dolutegravir in neurological outcomes. I have no conflict of interest. So as we all know, currently there is no effective treatment or disease-modifying therapy for HTLV1. The management is mainly symptomatic and unable to prevent progression. Some studies have shown that integrase inhibitors are capable of inhibiting HTLV ex vivo. However, in vivo studies have not shown a sustained suppression of proviral load or long-term clinical benefit. So we aimed to investigate the effects of dolutegravir use on neurological outcomes and to investigate functionality outcomes in people with HTLV1. This was a randomized, controlled, open-label pilot clinical study. The participants were randomized into dolutegravir group and into vitamin C group. We included adults, HTLV confirmed by Western blot or PCR, and we excluded all those with HIV, HCV, HBV, tuberculosis co-infection, concomitant use of corticosteroids, cancer, autoimmune disease, or welfares. So we evaluated participants in 2 moments at the baseline and after 24 weeks. All of those scales were applied to evaluate spasticity, functionality, strength, balance, pain, nocturia, and we also collected a blood sample at 2 moments. We use non-parametrical statistics for the analysis and to compare day 1 with week 24. And at the end, we included 83 participants. 43 into dolutegravir group and 40 into vitamin C group. After 24 weeks, we lost 5 participants in dolutegravir group and 5 participants in vitamin C group. As we can see in this table, we have a population mainly female sex, most of them with low scholarity, most of them with low income. And when we compare both of the groups, we can see that the vitamin C group was older. We didn't find any other differences between the groups. This table summarizes the main outcomes. As we can see here, we didn't find significant differences when we talk about pain, strength, nocturia, and CD8 cells. We found a borderline significance when we talk about trunk strength into dolutegravir participants after 24 weeks of the use of dolutegravir. But we found another interesting results. So we can see that after 24 weeks of the use of dolutegravir, this group improved the Ashworth spasticity scale. So we can see that after 24 of the use of dolutegravir, we have an improvement in spasticity, and we also have an improvement into the sensitivity score, mainly pinprick sensitivity. We also can see here that the dolutegravir group significantly decrease in proviral load, and we can see that after 24 weeks of the use of vitamin C, the vitamin C group had less CD4 cells count. The dolutegravir group have better functionality after 24 weeks of the use of dolutegravir, and both of the groups improved into the balance scores. But vitamin C group had worse scores into the TAC scores. These scores means that they had a higher risk of false score. In conclusion, in this pilot study, dolutegravir was associated with an improvement in functionality and in some neurological manifestations, such as spasticity and sensibility. Additionally, we found a significant reduction of proviral load after 24 weeks of treatment. Larger trials are necessary to define the role of dolutegravir in HTLV treatment. So I will want to acknowledge to the Fundação Baiana de Infectologia for supporting the project. I will thank to Brazilian Ministry of Health for providing dolutegravir, to my mentor, Dr. Carlos Brites, and to the LAPI research team. Thank you.
Thank you. The next speaker is Dr. Daisuke Kodama from Japan. The title is Molecular Targeted Agents for Treatment of HAM-TSP. Dr. Kodama.
Thank you, Chairman. We invented two agents for HAM, and there is no funding from any company. A background: there is no radical therapy for HAM. The motor disability progression of HAM creates proviral load. The PVL is maintained by proliferation and cell-to-cell infection. TAX is inappropriate for therapeutic target because the infected cells do not express TAX in vivo, however, express TAX after short culture in vitro. To explore target, we perform pathway analysis, and we investigated GLICAN. Using 12 subjects and samples, we performed microarray, and these are upregulated DEGs and downregulated DEGs, respectively. We identified 177 specific cellular genes. We define HAM-specific cellular genes as upregulated DEGs only in HAM. Mapping these genes to transcriptome database, we found HAM-specific pathways by chi-square test. we found significant harm-specific 12 specific pathways. 11 among 12 included AVL tyrosine kinase genes. The expression of AVL was verified by QRT-PCR. AVL is known a part of PCR AVL fused gene of B-cell origin, chronic myelogenous leukemia, CML. And tyrosine kinase inhibitor, TKI, were developed to treat CML. In previous report of AVL knockout mice, the thymocytes and T cells deaths were reported, and TKIs induced T cells to death. So our hypothesis is AVL may be a survival factor for HTLV infected cells, and TKIs may induce the infected cells to death. These were cell viability assay, and neoantigen and dasatinib induced the infected cell preferentially in drop symbols, and this is PBMC. For using the 80% of drug concentration for uninfected cells, dasatinib and neoantigen showed a significant reduction of PBL in live cells. Moreover, we found a concomitant case of HAM and CML who was administered imatinib for 30 months showed 84.2% reduction of PBL. In summary, we found AVL gene by pathway analysis, and TKIs reduce PBL in live cells. Therefore, AVL is a survival factor for HTLV infected cells. And we found HAM patient administered TKI without side effect. TKIs are potential therapeutic agent for HAM. Next, we searched glycan as the outermost structure related cell-to-cell cell-to-cell infection. But the role of glycan in cell-to-cell infection remains unknown. Using 12 subjects and membrane protein from that, lectin array was performed. Lectin means glycan protein that can bind specifically to glycan. The specific binding of glycan and lectin was detected on the surface of the lectin array. These are the results. The longitudinal axis and the transverse axis represent the LACNAC signal intensity and lectins. The black symbols indicate the lowest signal intensity in HAM among the groups. The red symbols indicate the highest signal in HAM among the groups. These are the kitchen-binding lectin, heaven family, LEL, tomato lectin, STL, potato lectin, and stinging nettle lectin, UDA. The common saccharide, common glycan specificity is N-acetyllactosamine. That's called LACNAC, in short. LACNAC is negative on normal lymphocyte. It's known. The expression of LACNAC was verified by lecturing ELISA. And the LACNAC biosynthesis gene has been identified as B3GNT2. And the expression was upregulated in harm. Using PVL data, PVL and B3GNT2 expression in infected individuals showed relatively strong correlation. LACNAC, to clarify the glycan at cell level, we perform flow cytometry using cell lines. The LACNAC signals are stronger in HTLV infected cells than uninfected cells. The ratio is shown in the parentheses. And the staining by UDA is better than by STL. The UDA stained cells shows LACNAC signal, UDA green, at the surface of the infected cell, at the plasma membrane. LACNAC gating by cadmium and CD4. The cadmium positive CD cells showed stronger LACNAC signals than in cadmium negative cells. Gating cadmium positive into low magenta and high green, the LACNAC signals stronger depending on the drug depending on the cadmium expression level. So our hypothesis, LACNAC may play a role in cell-to-cell infection. In infectivity assay, we use tax donor and recipient tax gel cell, jacket transfected with 3' UTR upstream of firefly luciferase gene. There is no relationship in the of the LACNAC in the infectivity assay system. The cell viability showed almost harmless until relatively high concentration. The white bars in infectivity assay shows the significant reduction of infectivity, especially UDA and 4F glucanac. 4F glucanac is a LACNAC metabolic inhibitor showed almost 80% of reduction in infectivity. And we performed conjugate formation assay using 4F glucanac. The pre-staining by dio green to infected cells and uninfected stained with dye red. And microscopic images were taken to count conjugate. In the combination of C19PL and MT2 with jacket shows the reduction of conjugates depending on the drug concentration. And in both combination, the conjugate formation rate or conjugate frequency shows a reduction depending on the concentration of 4F-glucanate. And conjugate formation rate and infectivity shows a very strong correlation each other. So in summary, N-acetyllactosamine LACNAC is specifically upregulated in HAM. And LACNAC is upregulated by HTLV infection via B3G and T2. And LACNAC biosynthesis metabolic inhibitor 4F-glucanate and LACNAC binding lectin STR and UDA suppress infectivity, and they are cell-to-cell infection inhibitor. And 4F-glucanate suppresses conjugate formation that creates infectivity. Thus, LACNAC is involved in HA1 cell-to-cell infection via conjugate formation and is a therapeutic target. And conclusions. A therapeutic target, we found two therapeutic targets, AVL and LACNAC. AVL is a survival factor, and TKIs induce cell death of infected cells. And LACNAC plays a role in cell-to-cell infection, and 4F-glucanate is an inhibitor. They are potential molecular targeted therapeutic agents for HAM and HA1 infection. Thank you for attention.
Thank you very much. Next presenter is Dr. Ashley Irons from Australia. The title is Chronic Inflammation and T-cell Exhaustion Ubiquitous in Central Australian HTLV-C infection is associated with defective proviruses retaining antisense HBZ gene. Okay, please.
All right, good morning, everyone. I would just like to acknowledge the First Nations participants from Mparntwe in Central Australia who participated in these studies. Now, Central Australia has some of the highest rates of HTLV-1 infection, and it's almost exclusively subtype C. One of the most common disease outcomes is HTLV-1 associated pulmonary disease, HAP-D. Life expectancy, like subtype A, is significantly less for those infected with subtype C, and this is particularly for high proviral loads. Now, subtype C is genetically divergent from subtype A, and this divergence is particularly focused in the PX region containing the regulatory genes. So we wanted to investigate what are the host and virus factors that contribute to early all-cause mortality and pulmonary disease, HAP-D. So we first set about characterizing the host immune response. So today I will focus on CD4 T cells, however we did also investigate CD8s. So there's been lots of previous studies of subtype A specific T cells, some of which detected high antigen-specific CD4s in HAM donors compared to asymptomatic carriers. So we stimulated PBMCs from Central Australia with taxon envelope peptide libraries. An important finding of this study was actually the significantly high levels of background activation detected in all of the CD4 phenotypes compared to negative controls. And we also found that lots of donors exhibited envelope and tax-specific responses. Interestingly, we found that the unstimulated Th1 frequency was negatively correlated to the proviral load, suggesting that infection might be downregulating differentiation of Th1. We found that the background activation of Th1 and the envelope-specific frequency, however, was positively correlated to the proviral load. So taken together, there's an overall dysregulation of the Th1 response. So we next looked at the host cytokine profile that might be produced from these dysfunctional T cells. Now, of course, the cytokine profile in subtype A is well studied. For HAM, there's a pro-inflammatory response, but for ATL, it's immunosuppressive. So we analysed the plasma of 77 individuals from Central Australia and performed logistic regression. And with the total cohort of an outcome of subtype C infection, we had a positive association of TNF-alpha, CXCL10, IL-2, IL-10 and age, while a negative association for interferon gamma and IL-8. When we looked at the positive donors only with an outcome of HAP-D, we found that IL-10, IL-6 and proviral load were positively associated, while again we saw that interferon gamma and IL-8 as negatively associated. So this is consistent with the Th1 dysregulation. We have IL-6, which inhibits the polarization of Th1 cells. IL-10 downregulates the expression of Th1 cytokines, and of course interferon gamma is one of these. We then investigated the CD4 phenotypes that are prominent in subtype C infection. So firstly, we found that there was an expansion of the effector memory, the TEM population. And then within this TEM population, we found 3 particular subsets that were enriched in subtype C infection. This was senescent TEM cells, chronically activated TEM cells, and then another population of chronic activation TEM cells, but HLA-DR negative. Notably, these 3 subsets were all enriched for T-cell exhaustion markers. So for our senescent cells, we had granzyme B, KLR-G1 and PD1. And for our non-senescent chronically activated cells, we had PD1, TIGIT and TIM3. So it's suggesting that T-cell exhaustion is playing an important role in subtype C infection and HAP-D. We then wanted to investigate the phenotype of HTLV1C infected cells and the provirus structure that is driving this dysfunctional response. So subtype A studies, among many, have identified CCR4 and FOXP3 as cells that are highly as cells that are highly infected and expanded. And for FOXP3, this is correlated to the proviral load. Now, we investigated these cells, and also given the high prevalence of pulmonary disease, we also looked at cells that may traffic to the lung. So we similarly found an expansion of the FOXP3 proxy phenotype, but importantly, there was an expansion in HTLV1C infection of the lung homeoproxy cells, and this was positively correlated to the proviral load. We also showed that these cells, these phenotypes are enriched for provirus, also including CCR4 negative cells. We also confirmed a significant chronic activation in these phenotypes. So we then asked, what is the structure of the provirus in these reservoirs that might be driving chronic inflammation? So we designed a limiting dilution single provirus amplification to amplify proviruses of 6 donors. And we sequenced using nanopore with the collaboration of Dr. Natasha Jans. So we recovered approximately 270 individual provirus sequences. We had around 3% that were full length. Around 20% were chimeric, so such that they had HTLV1 sequence with an internal human genome sequence. And around 75% contained large internal deletions. And as you can see here, these internal deletions were focused in the enzymatic and structural genes. And we confirmed this by droplet digital PCR. We then examined the proviral deletion breakpoints to see if there are any motifs. The most common breakpoints occurred at the LTR junctions, so we filtered all proviruses to remove those sequences, but surprisingly still found that the provirus breakpoints maintained LTR homology. We looked if there were any genomic features that were associated with pulmonary disease. So overall, HAP-D donors retain a higher level of genomic coverage, but importantly, at a nucleotide level, HAP-D donors retain the PX region, including the ability to express antisense HBZ at a much higher rate than non-HAP-D donors. We examined the provirus structure in the CD4 T cell phenotypes of interest. We found defective provirus in both the FOXP3 proxy and lung homing proxy cells. However, in the CCR4 negative cells, we found only full-length replication-competent provirus. So far, unfortunately, this is only in one donor. We're working on validating this in additional donors. But this may be a reservoir that maintains persistent infection and also may be a barrier to consider with anti-CCR4 treatments. Lastly, we examined these novel chimeric proviruses with the internal human genome sequence. Around 35% of these contain gene coding regions, while around 60% were centromeric. Now, these chimeric proviruses mapped across most of the host chromosomes. And the disruption of the host genome might result in aberrant gene expression alongside potentially the production of chimeric RNA and protein. So to conclude, we have shown that immune activation and exhaustion are defining characteristics of subtype C infection, and that's with a unique cytokine profile in the plasma. We have pathogenesis driven by a highly defective provirus landscape with HBZ gene retention, and the deletion breakpoints of the proviruses show LTR homology. Viral persistence is maintained by a replication-competent CCR4 negative reservoir. And lastly, chimeric proviruses may impact pathogenesis. So it's important to keep understanding the viral and host mechanisms that contribute to HTLV pathogenesis and disease, and so that we can develop effective and culturally appropriate therapeutic and preventative strategies. And thank you to everyone involved in these studies.
Thank you very much.
Thank you, Dr. Ashley. And we will go on then with next presentation about maladaptation to apolipoprotein BEC3G is linked to HTLV1 pathogenicity. I would like to invite Dr. Takafumi Shishiro from the Department of Hematology, Kumamoto University, Japan. Please.
Thank you for your kind introduction. Good morning, everyone. So today, I'd like to talk about association between adaptation to host antiviral factor and HTLV1 pathogenicity. My COA status is shown here. So as you know, HTLV1, HTLV2, HTLV1 belong to delta retroviruses and have similar provirus structure and regulative genes. However, there are some differences among these viruses. It is estimated that HTLV1 and HTLV2 originate from HTLV1 and HTLV2 respectively, but cross-species transmission of HTLV1 from monkey to human occurs more recently than that of HTLV2. Importantly, HTLV1 induces several diseases, including ATL and HAM, but HTLV2 and HTLV1 shows low pathogenicity in the host. So, to clarify the association of these primate retroviruses with their pathogenesis, the mutation rate and diversity of HTLV1, HTLV2, and STLV1 probables in infected individuals were analyzed by deep sequencing. Here you see the result, mutation detected in these HTLV1 carriers and HTLV2 infected individual and HTLV1 infected Japanese macaque. So x-axis shows the position of provirus and y-axis shows the percent of mutations. Compared to HTLV2 and STLV1, G2 mutations were highly observed in HTLV1 carriers. And focusing on the codon, majority of these mutations were found to be nonsense mutations. ApoX3G is a host antiretroviral factor, and during reverse transcription, ApoX3G protein induces C2U mutation in virus single-strand DNA. As a result, so G2 mutations were generated by ApoX3G in the provirus. When G2 mutation is occurred in TGG, stop codon will be generated by ApoX3G. Therefore, retroviruses usually adapt to the host by acquiring ApoX3G resistance, allowing for persistent infections. So we analyze impact of ApoX3G on mutations. We found that most of the mutations detected in HTLV1 carriers were preferred tag sequencing of ApoX3G, GG2AG, so suggesting that HTLV1 have not adapted to human ApoX3G. In contrast, HTLV2 and STLV1 are thought to have ApoX3G resistance. So to clarify the ApoX3G resistance mechanism of HTLV2, we created HIV virus particle overexpressing ApoX3G and these HTLV2 virus genes and evaluate the infectious efficiency of HIV by Lucifer's assay. So this is a result, and AP2 is an antisense protein of HTLV2 has human ApoX3G resistance like beef. So we're focusing on the antisense protein, and we found that HBZ and SBZ from Japanese macaque have a similar ApoX3G resistance, but HBZ did not have human ApoX3G resistance. Furthermore, so as you can see here, so antisense proteins bind to ApoX3Gs. So we found that AP2 SBZ is resistance to ApoX3G by binding to it, while HBZ binds to human ApoX3G but has no resistance. So based on the results so far, so we had new questions. First, so HTLV1 failed to adapt it to human ApoX3G and permit nonsense mutation in the provirus. But how do infected cells survive and proliferate in vivo? And second, human ApoX3G bind to HBZ. As you know, HBZ play important roles in HTLV1 infected cell survival and ATL oncogenesis. So how does human ApoX3 affect these HBZ functions? So we hypothesize that HTLV1 may be pathogenic because it is not adapted to human ApoX3G. So to test our hypothesis, we performed RT-qPCR, and ApoX3G was highly expressed in aggressive ATL, suggesting human ApoX3G is associated with ATL oncogenesis. So we knocked down human ApoX3 gene in ATL cell and performed RNA-seq analysis. And human ApoX3G was associated with TGF-beta signaling pathway. So to evaluate the activity of TGF-beta signaling pathway, we performed Lucifer's assay. Surprisingly, human ApoX3G enhanced the TGF-beta signaling pathway, as you can see here. And we previously reported, so HBZ and SBZ activate TGF-beta signaling pathway, Interestingly, so human apoptosis enhance TGF-beta signal pathway activated by HBG, as shown here. However, combination of human apoptosis and APA2 or simian apoptosis and SBG did not activate the TGF-beta signal pathway. So activation of the TGF-beta pathway by HBG and human apoptosis was suggested to be important in the mechanism of ATL oncogenesis. So we evaluate the two TGF-beta pathway inhibitor on the cell growth. Basically, generally, so activation TGF-beta pathway suppress cell proliferation of normal T cell. So as expected, so in HBV1 negative T cell line, both TGF-beta pathway inhibitor either promote or did not change the cell proliferation. However, in HBV1 positive infected cell and ATL cells, both TGF-beta pathway inhibitor suppress the cell proliferation, suggesting that activation of TGF-beta pathway is important for cell proliferation of ATL. So as you can see the figure, so we evaluate the ATL tumor growth in vivo using xenograft model. As expected, both TGF-beta pathway inhibitor suppress ATL tumor growth in vivo. So TGF-beta pathway inhibitor are novel therapeutic target for ATL. In conclusion, so HTLV1 and STLV1 had adapted to their host by counteracting apoptosis. However, human apoptosis induce hypermutation in HBV1 infected cell, resulting in inhibition of viral replication. So HBV1 is not fully adapted to human apoptosis. Interestingly, HBV1 use human apoptosis to enhance TGF-beta pathway activated by HBG, and activated TGF-beta pathway promotes cell proliferation of ATL. So misadaptation to HBV1 to human apoptosis is associated with ATL oncogenesis. So I'd like to thank my supervisor and the collaborators, and I'd like to close with a summary. Thank you for your attention.
Okay, so thank you. And we'll go on, and I think it will be possible, it's okay? Yes? To present, yes, the online work, and Dr. Zoran Vahid from Mashhad, University of Medical Science, Iran, the effect of teriflunomide on clinical signs, inflammatory and viral factors in patients with HAM.
Okay.
Yeah.
Good morning, ladies and gentlemen. My name is Zohra Vahid, a researcher in Research Center in Inflammatory Disease Derivation, HTLV Foundation, Mashhad University of Medical Science, Iran. I was very eager to attend the conference, but unfortunately my visa was rejected. I am very grateful to the HTLV 2024 Congress team and Professor Graham Tyler for giving me this opportunity to online present my paper. I would like to talk about the effect of teriflunomide on clinical science, inflammatory and viral factors in patients with HTLV1-induced myelopathy. Next slide. In relation to this presentation, I declare that I have no conflict of interests. HTLV1 is the first pathogenic human retrovirus and is associated with two primary diseases. The first disease, ATLL, and the second is HTLV1 associated myelopathy tropical spastic paraparesis, or HAM-TSP. HTLV1 prevalence in northeastern Iran, particularly in Mashhad, is estimated about 3%. HAM-TSP patients suffering from muscle weakness in the legs, stiffness of the limbs, difficulty walking, slow movement, and muscle spasms in the legs due to spinal cord disease. In addition, this patient usually present with neurogenic bladder disease. These clinical symptoms have been reported to be associated with increased HTLV1 proviral load. Pathophysiologically, there are high concentration of antibodies against HTLV1, both in the blood and the CSF. Next. The cells involved are mostly CD4 positive in the early stage of the disease and mostly CD8 positive in the late stage of the disease. Next slide, teriflunomide. Teriflunomide as an oral immunomodulator is approved for the treatment of autoimmune disease such as rheumatoid arthritis and multiple sclerosis. In terms of mechanism of action, it inhibits proliferation and reduces the number of activated T cell and B cells in CNS or in blood. Next slide, about a study design. I can say the present study was a triple-blind, randomized, placebo-controlled trial with a random sampling method with the aim of investigating the safety of teriflunomide in Iranian patients with HAM-TSP between March 2021 and June 2022. Patients referred to the Department of Neurology, HTLV1 Foundation, OM Hospital, Mashhad University of Medical Sciences, Iran, were selected from HTLV1 National Registry as participate for the study. Please, next slide. About exclusion criteria, I can say HTLV1 PCR positive, occurrence of symptom of spastic paraparesis, ruling out other causes of spastic paraparesis, motor disability score less than 9, patient consent to participate in the study, and I can say about exclusion criteria, the persons of other concomitant autoimmune disease that require receiving other immunosuppressive drug, lactating women not using reliable method of preventing pregnancy, women intending to get pregnant, also significant liver function disorders, kidney function disorder requiring hemodialysis. Please, next slide. The clinical examination data that were collected are as follows: demographic data, proviral load, tumor necrosis factor alpha as an inflammatory cytokine, time 25 football or T25FW as a clinically meaningful outcome measure for MS clinical trial, the Osame motor disability score or OMDS, urinary disturbance according to the urinary disturbance score or UDS, muscle strength using the Medical Research Council spasticity based on the modified Ashworth scale. Please, next slide. About treatment protocol, I can say patients in the intervention group receive 140 milligram teriflunomide tablet daily for 12 months, and patient in the control group receive a placebo tablet of the same color and shape as the intervention group daily for 12 months. Patients' clinical symptoms were assessed before and after treatment with teriflunomide and placebo for 12 months at 3 months intervals, 5 visits in total, by a neurologist using the T25FW, OMDS, UDS, MRC, and Ashworth criteria, as well as adverse effects. Please, next slide. About routine lab tests, I did biochemical blood tests, including AST, ALT, ALP, CBC, and so on. AST were determined periodic teriflunomide therapy, and AST, ALT, CBC, total bilirubin, and diuretic were determined in 1, 3, 6, 9, 12 months after the initiation of treatment. Please, next slide. You can see this slide about my results. 22 HAM-TSP patients who were referred to the HTLV clinic of Bayam Hospital's Medical Research Training Center were eligible and willing to participate, were included in the study. Patients were divided into 2 groups of 11, and the effect of teriflunomide was investigated. During the study, despite the necessary explanation and follow-up, 2 patients refused to continue taking the drug after 6 months. 1 patient from the intervention group due to the decision to become pregnant, and 1 patient from the control group due to her unwillingness to continue taking the drug. This next slide. The reduction rate was significant in the intervention group, p-value equal 0.01. In addition, reduction in walking time at the end of the study was significant between 2 groups, p-value equal 0.028. Please, next slide. Please, next. In the intervention group, in 45.5% of patients, a decrease of more than 20% in the T25FW test, which was 9.1% in the control group were observed. It should be noted that the presentation of reduction after treatment in the T25FW test was not significantly different between 2 groups. Please, next slide. About OMDS, you can see OMDS grades in the intervention group decreased from the beginning of the treatment till the end of treatment. In order to compare to trend of change in the severity of disability, the generalized linear mix model was used. The results of this model showed there was a significant difference between the 2 groups in the change in disability severity, p-value equal 0.001. Please, next slide. About UDS, I can say the total score of UDS in the intervention group decreased during the first 3 months after the treatment as compared to before the intervention. Then it slightly increased in the second 3 months and considerably decreased in the 3rd and 4th months. Please, next slide. UDS total test score before and 3, 6, 9, and 12 months after treatment with teriflunomide in HTLV patients. Additionally, the reduction in the UDS total score was significant in both groups. You can see in this slide, we have a decrease in UDS score. Next slide. There is no significant difference between the muscle strength in the intervention and control group at any time, and also about This next slide. There is no significant difference in spasticity between the intervention and control group at any time in Ashworth criteria. About HTLV1 coroviral load, according to this table, coroviral DNA load in the intervention group before the treatment was 176.1 plus minus 59.3 copy per milliliter, which decreased by 83.7 units in the 12 months after the treatment and reached 92.4 plus minus 60.1 copies per ml, which was statistically significant. P-value equal 0.003. This next slide. According to this figure, you can see HTLV1 coroviral load average before and after treatment with riflonomide in HTLV1 patients. We had magically decrease in this study. Please next slide. The levels of TNF alpha as inflammatory biomarker in the intervention group was 37.2 plus minus 40.9 before the intervention, and in the 12 months after the intervention reached 0.41 plus minus 8.8, which was not statistically significant. Please, next slide. About lab finding, the change in the biochemical tests, including AST, ALT, ALP, CBC, total bilirubin, and direct bilirubin during the 12 months treatment were not statistically significant. Please, next slide. We need treatment that modulate or are able to target the inflammatory response of the virus. Riflonomide is an immune system modulating drug that reduces the active lymphocytes of the central nervous system and exert anti-inflammatory and anti-proliferative effects. This study was the first evaluated the safety and efficacy of riflonomide for 1 year on clinical symptoms. Inflammatory and viral factors in patients with HTSB in northeastern Iran, an endemic area for HTLV1. Next slide. Our results showed that HTLV1 coroviral load decreased significantly in the intervention group compared to the control group. P-value less than 0.003. And also there was a significant difference between the two groups in the T25FW test in the intervention group compared to the control group, and also OMDS grades improved significantly in the intervention group compared to the control group, with p-value less than 0.001. Next slide. I think the latest slide. Thank you for your attention.
Thank you, everybody. Can we ask all the presenters to come to the front?
I think Dr. Bangan has a question. Yes?
Good morning. Great presentations, all the speakers. Congratulations. I have questions for Dr. Hirons, and I think I'm going to end asking a question to Dr. Sarkis. I'm from Peru, so I trained with Dr. Gotuso, who's there. One of the things that I learned when I was a med student in 2004 is looking at patterns of how HTLV1 was presenting, right? Strongyloides was very common, so strongyloides led to do HTLV1 testing. And then standardize a way in which we can clinically start seeing which patients may or not. Then we learn about scabies and other parasitic infections. We do see lung issues, but most of the time are not inflammatory. In nature are mainly like PJP pneumonia, tuberculosis, and other opportunistic infections. So the question that I mainly have is, because of lack of knowledge, honestly, is with HTLV1C being prevalent in Australia, how do you suspect on HTLV1 in a patient with a lung infection? Is like a standard that every time someone has some sort of like fibrosis like picture, is that HTLV1 is like mandatory as a screening? It's just like a general question for clinicians, I guess. The other thing is that within your studies, have you studied like, for the diagnosis, like sometimes we do for fibrosis in the lung, do you do lung biopsies? And if so, do you tend to do any studies within that, including like the markers that you can find? For example, my thesis actually was studying FOXP3 and Tregs in intestinal tissue of strongyloides, and we did find expansion of those cells in patients with hyperinfection for the strongyloides. We published that with Dr. Montes and Dr. Gotuso. And the other question I have, which is, I guess, for the both of you, is you are describing about this lung infection in HTLV1, but our Japanese colleagues have talked a lot about CAD-M1. And as you know, CAD-M1 is very frequent in lung tissue. But if we look at CAD-M1 even before we start knowing about strongyloides, there are several publications about being related to pulmonary fibrosis, related to asthma, bronchial diseases, but also to lung cancer. So I wonder if you have also looked into that into detail, because we have shown multiple times that CAD-M1 is actually overexpressed in patients with HTLV1 infection.
Thanks for the questions. Okay, so the first one about screening for HTLV1 in bronchiectasis or lung disease. So HTLV1's picked up because there's a lot of bloodstream co-infections and strongyloides, and of course lung disease, it'll be screened in sort of a wide variety of cases, or if they just present at the hospital for something else that could be associated. And the second question, second question was, the biopsies, yes. So thank you. The only lung samples that we are able to get is sputum samples. It would be great to get bowel or biopsies, but for us down in Melbourne, with the clinical samples we're able to get sent down, it's just plasma and PBMCs, but perhaps an animal model is useful in that scenario. And lastly, for CAD-M1, yes, we'd really like to look at that expression on these cells. There's only so many things we can look at in a small sample, but yes, it's an important marker to look at.
In the lung of the animals, we didn't look for that marker, so no. Maybe, but for the HTLV, because now we are running another study for HTLV1A, and the study is still ongoing, so we might add this marker on our panel and look for that, for the HTLV1A. But for the AC, we didn't.
Okay, so Dr. Bangham, please.
More
Thank
for
you.
Ashley Hirons. Your observation of the very high frequency of internal deletions is extremely unusual. Even odder is the association with CCR4 negative and CCR4 positive cells. So the first question, can you exclude an artifact in selecting the cells for identification or sequencing, which might have led to an over-representation of deleted genomes?
So the method, there's caveats to every sequencing method. So it was a limiting dilution. So there was less than 30% positive wells for a single provirus. So it gives it a much higher probability that it came from a single amplification. So there would be no biases in terms of if there's one copy in the well, it's going to be amplified, whether it's defective or full length.
Okay. If it's true, then perhaps the most likely explanation is immune selection for deletion of the plus strand, particularly because you get maintenance of HBZ expression, as you might expect in the continuous maintained clones. But the odd thing is that it's associated with CCR4 negativity, and yet it's known that HBZ can induce expression of CCR4, so it's the wrong way around. So how do you explain this?
Yeah, it's definitely, it was an unexpected result that we found full-length provirus in CCR4 negative cells.
Negative.
That was an, negative control I thought it would be.
I mean, maybe it's the CCR4 negative ones that don't express the plus strand, and they don't get selected.
Yes, it's very hypothetical. My thoughts were similar, and also that the CCR4 positive cells, which have internal deletions, are likely, trafficking to sites of information, so they're exposing, and then they're getting the negative cells are getting selected for.
Thank you. Next.
Hi, good morning. My question is for Iris Montaño. I think your data from this trial are very important. So I think is the proof or not proof that in clinical practice in vivo, dolutegravir will work. You presented data about the average reduction at 24 weeks in 38 patients taking dolutegravir, but in other viruses, in hepatitis B, hepatitis C, or HIV, we try to provide also individual declines. So it's important always to try to show what significant reduction in provider load, and this is beyond 0.5 logs divided by 3, what proportion of patients show that. And there is no biological fluctuation or problems with reproducibility of the methodological assay. Can you report what proportion of patients show a significant decline in viral load? Was for her.
Just to understand better the question, you, sir, said that if I know what kind of patients show a significant decrease in provider load, if we did that analysis?
That will be that the drug is providing a direct effect on replication, okay? Otherwise, will be biological fluctuation or variability in the diagnostic assay you are using. You provide the average reduction in 38 patients, but how many of them individually show a significant decline?
We still didn't do that analysis, but maybe we will have, because we didn't only made two. We didn't. I showed in data that we assessed provider load at 2 times, at the baseline and also at 24 weeks. But we also collected another blood samples, and maybe, not now, but we will do that analysis to see fluctuations on provider load that it is expected to be. We will do that analysis to know what kind of patients had a better response to dolutegravir. We still didn't have that answer.
Okay, thank you. Now.
Yeah, I would like to make a comment and particularly talk with the people that showed that there is a decrease in the provirus load with the different treatment that we have seen today. So this is more a discussion, I think, for the field. Why do we see a decrease in the virus load using an integrase inhibitor when we do believe that you have probably expansion of the infected cells? So if you decrease the virus load, you must be killing also the cells that were carrying the virus at the provirus, and then blocking also reinfection. So this suggests to me, your data, that there is a much more dynamic viral replication occurring in HTLV than we think. Because otherwise you would not expect to see it so relatively fast. I would like you to comment on this. Explain what you think is happening.
I think, do you want to answer? No, yeah.
Maybe.
I'll make a comment, Fefa. We've been saying for years that it goes on. There's nothing that changes. The virus has not changed. It's still trying to transmit. But it's reached an equilibrium with an efficient immune response. So most of the ones that are trying to transmit, to infect new clones, get killed before they can complete it. But we've got evidence, and we've modeled this as well. So it's continuing all the time. Yeah. Yeah, there's turnover, certainly.
Turnover,
yeah. Well, about time too.
Okay.
Okay, so although there are many questions from through the internet, but time is extended. So shall we close this session? Thank you very much.
So only one question. Please, a moment. We have here a closing announcement. Next up, after the coffee break, is the penultimate section on persistence. Okay, thank you.